Welcome back, guys. In this video, we're going to go through the anatomy and physiology of accessory organs in the digestive tract, which for our case is going to mean liver, pancreas, and gallbladder. So as a first overview, the anatomy of this is fairly straightforward. The liver produces bile, and the pancreas produces a fluid that someone decided should be called pancreatic juice. And those secretions eventually need to wind up in the small intestine. So in our picture, this is the small intestine, right? Specifically the duodenum. Remember, chyme, the contents from the stomach would have emptied from above. They're just not actually drawing the stomach on this view. And so the big picture of this is we need to get bile from the liver and pancreatic juice from the pancreas into the small intestine at the right time. And the way this works is the liver produces bile, and when you're not digesting a meal, that bile makes its way into the gallbladder. Okay, so this shows a slightly better view. These hepatic ducts are draining bile away from the liver where it's being produced. And if you're not digesting a meal, that bile will route up this cystic duct. The cystic duct is just the connector to the gallbladder. And the gallbladder doesn't really do a whole lot of anything. It's just a pouch. And so it's a container to store bile in between meals. And so when you're not eating, the liver fills the gallbladder up full of bile through the cystic duct. And then when you do eat a meal, the bile from the gallbladder will be pushed out by a contraction. So back out the cystic duct. But now this time we're going to go down the common bile duct, which is eventually going to lead to the small intestine. Right? So if we zoom in right here, this is the common bile duct coming down from the gallbladder and the liver, entering the small intestine. Your bile will be squeezed into the small intestine at that point. It's also the same shared opening that the pancreas has, right, through this pancreatic duct. Okay, and so the gallbladder's common hepatic duct, common bile duct, and the pancreas's pancreatic duct, they merge together and squeeze bile and pancreatic juice, which is going to contain digestive enzymes, bicarbs, so there'll be bases in pancreatic juice to neutralize that stomach acid and better digest nutrients, because the small intestine is where we expect most of our nutrient absorption to happen. Right. So in all of this, we need to talk a little bit about function of the liver and its odd blood flow setup. So a lot of people refer to the liver as a filter, but it really isn't a filter at all. The liver is more like a metabolic organ. So your liver is full of enzymes that just convert harmful or toxic things into a chemical form that's easier for your kidneys to get rid of. Okay, and so we want to get anything that comes in from the digestive system that isn't supposed to be there, like toxins or poisons, to the liver immediately if we possibly can. So the way that this works is most organs have an artery bringing blood in and a vein leaving to go back to the heart, and that's it. The liver has a weird additional structure called the hepatic portal vein, and a portal vein is just something that links one organ directly to another without going to the heart first. And so to better make sense of this, I made a drawing with my admittedly horrible art skills that illustrate this. So if this is our heart, blood is coming up and out the aorta. Your aorta delivers blood to every organ in the body other than the lungs. And so I'm showing only two branches here. The aorta will branch into the mesenteric arteries that deliver blood to your GI organs. It also has an artery going to the liver that we call the hepatic artery. That's necessary because, remember, blood in your arteries has two things that you really care about. It has lots of oxygen, and arteries also have high pressure. The pressure is what's keeping that blood moving, because we want to deliver the oxygen to the GI organs, to the liver, to every organ in the body. Now, what would normally happen here is, if the liver was typical, the GI organs, their vein would connect back around and eventually to the vena cava, because the vena cava returns blood back to the heart. That doesn't actually happen, though. So the vein coming off of your GI organs, that is the portal vein, and it drains directly to the liver. Okay, so this guy would be your hepatic portal vein. Most people just call it a portal vein because it's the only major portal vein in the body, but technically hepatic portal vein. So your liver really has two separate inputs. It has the hepatic artery that's bringing lots of oxygen and pressure into the liver that it needs to survive and for blood to keep flowing. And it also has the portal vein, which you expect to be pretty low in oxygen and pressure because it already went through the GI organs, right, like the stomach and the intestines. But what you expect the portal vein to have is high concentrations of both nutrients, like glucose, that the liver can store as glycogen, and potentially high concentrations of toxins that the liver is supposed to metabolize. And so as these toxins, which can also include medications, right, when they go through the liver, right, and then eventually all of that blood from both the hepatic artery and the portal vein will leave the liver through the hepatic veins. As the liver metabolizes and removes those things from the blood, we call that the first pass effect. Right? First pass because it's the first passage of blood through the liver to expose those toxins or medications or drugs to enzymes in the liver that are going to convert them into a safer form. Right? And whatever comes out on the other side, that will go back to the vena cava, back to the heart, and enter general circulation. Okay, so some drugs have a very, very strong first pass effect, and they're almost all gone by the time you get out of the liver. Those drugs have to be administered a different way other than orally, so they don't go through the portal vein. And then for some other drugs, the first pass effect is fairly minor, and it's just not that big of a deal. Right? But this graphic is showing you all the different veins that drain back to the portal vein. So blood flow from the spleen, splenic vein, inferior mesenteric and superior mesenteric are coming from the intestines, and the gastric artery from the stomach. These are all feeding into that portal vein, going through the liver first before you can go back to general circulation in the vena cava and back to the heart. All right, to get details in what's going on in the liver, we need to look at it under a microscope. So if you put liver tissue under a scope, you see this hexagon pattern. And each one of these hexagon-shaped structures we call a lobule. And the way it works is every corner of the lobule, you can't really see them very well here, but they all have three different structures. They're really tiny, but you would have a hepatic arteriole, which is a branch off of the hepatic artery. You have a venule, right, often called a portal venule. Right? And sometimes this is also called a portal arteriole, either way. And then you also have a third tube that is a bile duct. Right? So at every corner around the whole lobule, you're going to have those three things. We call those in combination a portal triad. Triad because there are three of them. Right? And blood from both the portal venule and from the portal or hepatic arteriole, they're going to flow towards the center. And what you're seeing is all these little cracks in the tissue that you notice, they're all feeding blood towards the center. This middle structure we call a central vein. That central vein is what's going to take blood up and out of the liver to leave, right, and enter the hepatic veins. And so just remember, a mixture of blood from arterioles and venules is flowing towards the center from every corner of the lobule, every portal triad. Conversely, bile is flowing out towards the bile duct, right? And so bile and blood are supposed to be flowing opposite ways so that blood moves past all these hepatocytes, right? If you zoom in as close as possible, you can't really see them clearly, but all of these individual cells that are throughout the liver, those are just hepatocytes. Those are where you find the enzymes that are going to be detoxifying blood, removing molecules that aren't supposed to be there. And we just want our blood vessels to move past those hepatocytes so that the cleaned blood can enter the central vein and leave the liver. All right, so in an illustrated or animated form, it looks like this, right? Portal venule, portal arteriole, bile duct, right? These things combined make up one of your portal triads. These sinusoids sinusoid is a special name for the capillaries of the liver. They get a unique name because they're odd-looking capillaries. They have a bunch of holes in the capillary wall that makes it easy for substances to diffuse in and out of the capillary. And so as the blood moves from portal triad towards the central vein, you expect toxins, waste products, anything that the liver needs to metabolize, along with nutrients like glucose, to diffuse out of the blood, enter the liver. And then the blood that's left once you get to the central vein, that is going to wind up leaving through the hepatic veins, which eventually make their way to the vena cava, like we talked about before, right? And of course, that blood keeps circulating around over and over again. But the portal venule, remember, that's where you're getting your first pass, right? Blood coming in from the GI tract. And the liver is full of thousands of these lobules, all working in conjunction with each other. That's why it's possible for the liver to regenerate, is that each lobule kind of does its own thing. And if you damage a small portion of the liver, the entire function isn't lost, and it can regrow and replace itself over time. Okay, so now that we've got the anatomy figured out, let's add a few extra things on the end. One is the actual function of bile. We already said that the liver produces bile, which gets stored in the gallbladder and eventually moves into the small intestine after the stomach is done digesting. What bile contains are a handful of things. One is it has a slightly alkaline or basic pH. The purpose of that is to neutralize acids coming from the stomach. That's pretty straightforward. You also have something called bile salts, which are amphipathic molecules. And amphipathic really just means that they're a mixture of hydrophobic and hydrophilic. They act much like soaps do. Even dish soap is an amphipathic molecule. The purpose is we want those bile salts to break apart or emulsify fats in food. Fat droplets, just like if you mix oil and water in your kitchen, they form these big globules that are too large to be taken in by endocytosis. And so bile salts are really just acting like soaps to break apart the fat globule into smaller and smaller pieces. Those tiny little fat particles are eventually called micelles. And these are not moved by active transport or diffusion. They're moved by endocytosis. So this intestinal epithelial cell will grab onto a micelle, bring it into its cytoplasm, and eventually push those micelles out the other side. And those fats are moved into circulation not through arteries and veins, but through the lymphatic system. Now, this lacteal, that's just a capillary of the lymphatic system. Eventually, those lacteals will drain back into blood as you enter the heart. And so fats end up in the blood supply. They just take an indirect route through the lymphatic system. Right. The last function of bile is removal of waste products. Specifically, the liver is a big part of the cleanup process for dead red blood cells. And so there's a waste product called bilirubin that is the breakdown product from heme. That's part of the hemoglobin molecule in red blood cells. That's what gives bile its greenish-yellow color. It's also what accumulates in the blood of people with jaundice that aren't able to eliminate bile, and so the bilirubin builds up in their blood instead. All right. Now that we've got liver taken care of, a brief detour to talk pancreas. The pancreas secretes an exocrine substance that someone named pancreatic juice, which sounds odd, but that pancreatic juice is pretty straightforward. It's an exocrine secretion. It drains down these tubes that become what we call the pancreatic duct, and it enters the small intestine, remember, at the exact same location that the bile duct comes from. Right? This would be gallbladder up here. So bile and pancreatic juice enter the small intestine directly, and the pancreas is mostly making bicarb, which, remember, is a base, so it neutralizes stomach acid. And then all of these enzymes, amylases, lipases, remember, amylases metabolize starch, lipases for lipids, nucleases for nucleic acids like RNA and DNA, and this is where we're getting a lot of the breakdown of our food into individual molecules. Also proteases to digest proteins. Just like the stomach, these are secreted in an inactive form, and then they're activated once they get to the small intestine. Right? Just make sure in your mind you separate this from the endocrine function of the pancreas that we saw previously. Anything in a pancreatic islet, that is endocrine. That's alpha and beta cells secreting insulin and glucagon. these acinar cells, so when they call this an exocrine acinus, acinar cells are the ones that are secreting pancreatic juice. That's the exocrine portion. It's entirely digestive. All right. Okay, last idea in all of this. These organs, like the liver and pancreas and gallbladder, they have to be told when they're supposed to function. And what I mean by that is there's no reason that these organs should be active all the time. They only need to really do their thing when there's food present in the small intestine. And the way that we signal that the liver and gallbladder and pancreas are supposed to be activated is through two hormones called CCK and secretin, or some people call it secretin. And the idea is CCK and secretin are produced in the small intestine, but only when there's food present. So when the stomach empties into the small intestine, the epithelial cells of the small intestine release CCK and secretin, and those hormones have target cells 4 different places. On the liver, which increases the production of bile. On the gallbladder, which stimulates contraction, right, to push stored bile out to the small intestine. In the pancreas, to increase production of pancreatic juice. And this hepatopancreatic sphincter, that is the sphincter that's found at the opening where the pancreatic duct and the bile duct open into the small intestine. We have to relax that sphincter to allow those fluids to move past, enter small intestine. Okay, so this is all about timing. We want to get these digestive enzymes and bicarb from bile and pancreatic juice into the small intestine, but only right after the stomach empties. CCK and secretin are just communication, right? How the small intestine tells those organs that now is the time to make those functions happen. All right. As always, hope this was helpful. Follow along in the playlist because things make more sense when you follow them in order. And the next video is actually going to be over taste and smell, which is really sort of a hybrid of nervous system and digestive system, and hopefully we'll make it all come together. So subscribe to the channel so you get those updates, and look for the next video to come out soon.